Good morning, everyone. Today we are going to discuss the fourth module of molecular basis of inheritance. In this, we are going to discuss the transcription. So, last class we have studied about DNA replication, and in this chapter, we are going to discuss the transcription. So, what do you mean by transcription? Since the DNA is always located within the nucleus and it is the carbon copy of an organism, that means all the characteristics what a person exhibits, that is encoded in the base sequence pattern, and that is the only the detailed characteristics in an encoded manner safely it is located within the nucleus. And the size wise also DNA molecule is larger, and the length if we are saying that is 2.2 meters, and the base we are saying means that is haploid set means 3.3 into 10 to the power of 9 base. So here that much of large molecule, and it is the only carbon copy of the organism, so it always remains in the nucleus. It cannot go out because of its large size through the nuclear pore. So they will reside within the nucleus itself, whereas the protein synthesis or the expression of characters is mainly in the form of proteins. So that process is taking place within the cytoplasm. So this information from the nucleus will have to carry into the cytoplasm, and according to that instructions the protein has to be produced, and that protein is only expressed in the form of varied characteristics. So how this translation is possible? So it is mediated with another type of nucleotide chains. That chains collectively we'll call it as RNA. So the transcription means the information that is encoded within the DNA is transcribed, is copied in the form of into another form, that is in the form of RNA, and these RNA can pass through the nuclear pore and they can take initiative for the process of protein synthesis, or else we can say translation. So transcription means it is a process of copying of genetic information from one strand of DNA into RNA. So why we say the one strand? Because RNA, the nature is single stranded, whereas DNA is double stranded. So out of that double stranded DNA, any one of the strand only should act as the template. And from that template only the complementary base pairing takes place and the synthesis of RNA is taking place. So here from a double stranded RNA, a single strand, the DNA, a single stranded RNA is formed, and that process is called as transcription. So it is mediated with the help of an enzyme. That enzyme is called as DNA dependent RNA polymerase. DNA dependent RNA polymerase. That is, three types are there, because within the cell itself there are three types of RNAs are there: messenger RNA, transfer RNA, and ribosomal RNA. So here we can explain that one with the help of a mnemonics RMT. That means RNA polymerase 1 is for ribosomal RNA, R, and the RNA polymerase 2 is for messenger RNA, second letter M. Then RNA polymerase 3 is for transfer RNA. So RMT. So that order of polymerase enzyme we can easily remember. RMT stands for RNA polymerase 1, 2, and 3. 1 is for ribosomal RNA, 2 is for messenger RNA, and 3 is for transfer RNA. Then here also as same like, there is the same like the DNA polymerase, DNA replication, unwinding of DNA chain. And one striking dissimilarity is there both the strands are acting as the template, means here only one strand is acting as the template. And the complementary base pairing takes place. But only one difference, that is wherever U in the template, wherever A in the template, instead of forming complementary base pair with thymine, here in RNA there is uracil. So A binds with uracil. There is no thymine in RNA. So all rest all processes same. So base pairing, only one base pairing, only small slightly different, that is adenine always binds with uracil. Then enzymes, when you are saying already I said 3 types of enzymes RMT. Then each enzyme again it is made up of subunits. There are 5 subunits plus sigma factor. That means 2 alpha component, 1 beta, then 1 beta dash or beta prime, then omega. So 5 plus 1 initiation factor, that is sigma factor we can say. All these things together forms the holoenzyme component of RNA polymerase enzyme. And this sigma factor is required only for the initiation. As soon as the initiation takes place, the sigma factor detaches and that remaining components together we'll call it as coenzyme. So coenzyme plus sigma factor forms the holoenzyme. The holoenzyme is required for the initiation and immediately after that sigma factor detaches and this coenzyme component of the RNAP takes initiative for the elongation of the transcription. Then here we can say this shows the heterocatalytic. So DNA replication is autocatalytic, means RNA production is heterocatalytic. That means from a single-stranded DNA, from a double-stranded DNA molecule, the formation of a single-stranded RNA mediated with the DNA-dependent RNA polymerase. So DNA takes initiative for the formation of dissimilar molecule. DNA replication, it is similar molecule, means here it is dissimilar molecule, that is RNA. And the direction is always 5-3 direction. That means the strand which is having the 3-5 direction, the template strand will be decided by the sequences. And that strand which is having the 3-5 direction will act as the template for the newly synthesized RNA. Now we'll move on to the next slide. So here the question naturally arises, why the DNA, both the strands of the DNA, is not taking part in the transcription. One of the reason, RNA generally it is single stranded. So for that particular moment we need only one single stranded RNA that can be formed from a single stranded DNA. So if both the strands are acting as the template, they will code for 2 RNA molecules. Code for 2 RNA molecules, which will show the different sequences because both the DNA is anti-parallel and they show the complementary base pairs. So if both of them they are acting as the template, means the formation of RNA molecule will be different. And if they code for proteins, the sequence of amino acids also will be different. So we won't get that particular moment which protein we require, only one RNA only can supply that particular type of protein. If any complementary RNA is forming, means the protein which is formed from that RNA will be entirely different, which will be accumulated and it will be treated as a waste. So even if both the DNA is acting as template, that particular moment we need only one RNA and one type of protein. So unwantedly there will be an excess amount of another protein which is treated as the waste. So that particular moment one RNA, that from that one type of protein only we need required. So we need only one DNA strand. Then even if two RNAs are formed, suppose, since that RNA carry the complementary bases, they will have a chance to form double-stranded RNA. So whenever this double-stranded RNA is forming, that automatically stops the synthesis of protein synthesis, because protein synthesis is mediated with the codon-anticodon sequence of rRNA and tRNA. So there the base pairing will not take place, because the mRNA itself, which is formed as double-stranded, means the translation will not take place. So that means that will come to an end. No protein synthesis will take place. So all these are the reasons why both the strands of DNA is not taking part in the formation of RNA. Then there are small differences between the transcription process in prokaryotes and eukaryotes. So we will deal separately. In prokaryotes, the process of transcription is completed within the nucleoid region, because there is no definite nucleus. And the phase when we are saying the transcription in both in prokaryotes as well as eukaryotes. Eukaryotes, when you are saying it is in nucleus itself. The RNA synthesis is taking place within the nucleus, and the phases when you are saying G1 and G2 of interphase stage. DNA synthesis in S1 means transcription in G1 and G2 phase, and in prokaryote it is in nucleoid region, because there is no definite nucleus. Then prokaryote, there is one more difference. That is, the same DNA is acting as the template, and at the same time, from the same sequence of genic sequence, that particular region will take part for the synthesis of more than one mRNA. Hence it is called as polysystronic, which will take up initiative for the synthesis of more than one polypeptide. So one DNA which can produce different types of mRNA, and that mRNA can take part in the synthesis of many proteins. So one strand itself, many proteins at a time, if they are taking part, then that type of this one is called as polysystronic. So prokaryotic DNA is polysystronic, or RNA is polysystronic, whereas eukaryotes are monosystronic. That means one RNA which will have its own promoter, then its own terminator. From that region only one polypeptide only will be produced at a time. So in prokaryotes it is polysystronic, eukaryotes it is monosystronic. One polypeptide at a time from an mRNA, which in turn from a single-stranded DNA. Now we will move on to the initiation. So as usual, there also there is a particular region. That region is called as promoter, the site where the RNA polymerase binds and starts the initiation of transcription is called as promoter. So promoter is a sequence, a special type of sequence which can be recognized by the cellular machinery. And because of that special sequence, RNA primer, RNA polymerase can bind at that particular site. So before that transcription region, there are many repeating sequences, UTR, untranslated region, UTR, or transcriptionally inactive region. So some repetitive sequences will be there. Even after the terminator also, there are many repetitive sequences. So based on that sequence only, they will recognize that is 10 bases before or 35 bases after. There can be presence of initiator site and the terminator site. So here, with the help of helicase and other topoisomerase and all, the unwinding can take place. And as a result, the unzipping of the nitrogen base pairs. So there will be a formation of transcription bubble at the initiator site. And to that initiator site, or to the promoter. That means it is the site on the DNA which allows the binding of RNA polymerase. So here we have to specifically say that is the promoter region is seen on the template strand. The template strand will be having the polarity of 3-5 direction. Then only the new synthesized strand will be 5-3 direction. So at the promoter site, the location is at the third end of the template strand, which allows the binding of RNA polymerase, including the sigma factor at the promoter site. So as soon as the binding takes place, the base sequences will complementary base sequence. The difference is that when they form the base pairing, the nucleotides will be ribonucleotides. In our DNA replication, it is deoxyribonucleotides, means here the base pairing takes place between the ribonucleotides. That is why in the template, if it is A, means the newly synthesized RNA strand, it will pair up with U. So there is no T. So that is the only difference. With the help of energy and all, the nucleotide elongation can takes place. So initiation, we require initiation factor, proteinaceous factor, that is the sigma factor attachment at the promoter site. Not only sigma factor, RNA polymerase along with the sigma factor will bind, that results in unbinding of the double helix. And one of the strand that is called as template with a polarity 3-5 will act as the template. And as soon as the initiation taken place, means sigma factor will detach. That is approximately 20 base pairing takes place, means immediately the sigma factor detaches. Then the elongation factors binds. With the help of elongation factors, RNA polymerase base pairing will proceed in the direction towards the third end of the new strand, or we can say 5 end of the template strand. And here the sigma factor already it is detached. The remaining part of the core enzyme of the RNA is mediating the elongation. And we'll move on to the termination. So when it reaches the terminator site, there will be a proteinaceous factor that is called as Rho factor. Rho factor will bind at the terminator site, which will decrease the affinity of, or which will stop the affinity of binding tendency of RNA polymerase, and as a result RNA polymerase will detach. So by the time we will get within that transcription bubble the long stretch of ribonucleotide strand, or we can say an RNA molecule. So once this Rho factor detaches, the formed RNA, newly formed RNA also will be peeled out of the transcription bubble, and the helix reformation will take place. So here we will move a little more detailed structure about the transcription unit. So I said it is a region which takes part, region in the DNA which takes part in the transcription is called as transcription unit, which consists of a promoter, the structural gene, and a terminator. And out of these two strands of DNA, one strand will act as the template, which is having the polarity 3-5 direction. And the other strand, which do not take part in transcription, is called as non-template or coding strand. Why it is called as coding strand means when from the DNA template, that is the strand with 3-5 polarity, when they form the RNA, which is in the direction of 5-3, that code sequences in the RNA will be exactly same to the non-template strand, except instead of T, there will be only U. So the base sequences in the newly synthesized mRNA will be exactly the same as the non-template strand, hence that Non-template strand is called as coding strand. Then the specificity of promoter. It is the site which is located towards the 5' end of the structural gene, and it allows the binding of RNA polymerase and starts the transcription. Then the terminator means the terminator is the site which allows the detachment of RNA polymerase after the transcription, that is mediated with the help of Rho factor. So that site where the transcription comes to an end, that is called as terminator. So the region of sequences between the promoter and the terminator is collectively called as structural gene. So what do you mean by structural gene? Means it is the region from where the protein synthesis or that base sequence is based on that base sequences only the amino acid sequence will be determined. So the region which actively take part in the protein synthesis from the mRNA, that region is called as structural gene. Now we'll move on to the diagrammatic figure. So here two strands are given. One strand is named as template, and in that itself three regions: promoter, structural gene, and terminator. So promoter is located at the third end of the template strand, whereas terminator is located at the 5 end of the template strand. The strand which do not take part in transcription is called as coding strand or non-template strand. So this is the diagrammatic figure of transcription in prokaryotes. The first step is the initiation. So here you can see in the double-stranded DNA. The DNA strand with polarity 3 to 5 direction. At the third end, that is marked as yellow color, that is the promoter region, which allows the RNA polymerase binding, including the sigma factor. And as well as a result of binding, the complementary base pairing with the ribonucleotides takes place. And the base pairing, temporary base pairing, takes place with the template strand and the free nucleotides. And as the process elongates, second step, elongation, detachment of the sigma factor, and the transcription bubbles move from one region to another region with the help of RNA core enzyme of RNA polymerase, that means excluding the sigma factor. The last step is termination. When this reaches the terminator site, that means 5 end of the template strand, there the terminator signal will be there, that allows the binding of Rho factor on the terminator site. So this will result in less affinity of RNA polymerase to bind with the DNA template. So the RNA polymerase will detaches, and the newly synthesized RNA also will be peeled out of the replication transcription bubble, and the two strands of the DNA will be free. Since they are complementary, the helix reformation takes place. There, the transcription comes to an end. But there are certain factors also we have to summarize how the recognition and initiation is taking place. The first point when you have to write unwinding of helix. And one of the strand will act as the template, the other one is acting as the non-template. And initiation is taking place mediated with the help of RNA polymerase, which binds at the third end, that is at the promoter site, along with the sigma factor. Then transcription bubble forms, complementary base pairing, that is ribonucleotide base pairing, So instead of uracil, thymine is base pair. And the direction of formation of new synthesis is always 5 to 3 direction. And as soon as the initiation taken place, the detachment of the sigma factor approximately after 7 to 10 base pairs, maximum 20 base pairs. And immediately the detachment of sigma factor takes place. The elongation, with the help of elongation factor in the 5-3 direction of the new strand, it will continue by the base pairing complexes. Then this process will come to an end when it reaches the terminator. There the termination factors and releasing factors will bind, that will result in, pro factor will say, that will result in detachment of the RNA polymerase. Then the newly synthesized, or we can say the nascent RNA, will peel out of the site of transcription, detachment of RNA polymerase, and the template strand and the coding strand wind up together and they forms the helix, or we can say helix reformation. Now we'll move on to, there are small changes in the eukaryotes. So eukaryotes, these RNAs which are formed, which will modify post-transcriptional modifications, we can say. So the first formed RNA, we'll call it as heteronuclear RNA. It is in the nucleus. From there, it has to pass on to the cytoplasm, because eukaryotes means there is a distinct compartmentalization between nucleus and the cytoplasm. Whereas in prokaryotes, there is no compartmentalization, just a nucleoid region, and there itself the translation will start. So transcription and translation is coupled together within the prokaryotes because there is no compartmentalization. But here, from the nucleus, this RNAs which are formed according to the RNA polymerase RMT, I said, 1 means ribosomal RNA, 2 means messenger RNA, 3 means transfer RNA. So accordingly that has to be modified. So how this maturation of RNA from hnRNA to messenger RNA is taking place, that is called as post-transcriptional modification. Because it is passing on to the cytoplasm, there more enzymatic activity, more degradation chances are there for the RNA. So it has to be protected well. So the maturation step or the post-transcriptional step includes 3 headings: capping, tailing, and splicing. So the first step we'll call it as capping. That is the newly synthesized hnRNA at the 5 end. At the 5 end, the attachment of 7-methyl guanosine triphosphate. With the help of 7-methyl guanosine triphosphate will be attached at the 5 end of the hnRNA, and that forms the cap. So that process is called as capping. The same way at the 3rd end of the hnRNA, there is approximately 200 molecules of adenosine triphosphate will be attached one after another. That forms a long tail, polyadenylation we'll say. That step is called as polyadenylation. Poly-A tail will be formed. Then in between there are certain sequences, that is coding sequences and non-coding sequences, or we can say expressible region and intervening region. That means certain regions, certain nucleotide base pairs takes part in the translation, which will be expressed in the form of amino acid sequences, or which take part in the protein synthesis. That region is called as exons, expressible. It will be expressed in the form of polypeptide. But in between that, there are certain junk genes, we can say, which is intervening sequences, which do not take part in the protein synthesis. That sequences are called as introns. These introns are formed as a result of evolution. When we ourselves are the product of evolution. So from single unicellular bacteria to most complex human beings, there are many genes which are gradually became functionless and non-coding because many advanced organ systems and all are formed. So primitive functions and all lost in the process of evolution. So as a result, that regions also have become functionless. So that non-coding sequences are collectively called as introns. So these introns have to be removed and the exons have to be united. That process is called as splicing. So the splicing means the formation of continuous exons or joining of exons together after the removal of intron. So how this is done? That is done with the help of a special type of RNA that is called as small nuclear ribonucleoproteins plus snRNA, small nuclear RNA. So small nuclear ribonucleoproteins, NURBs we'll say, with the help of proteins, as well as a small stretch of RNA. So that is collectively called as spliceosome. So one end of the spliceosome will attach at the 5' end of the intron. The other end of the RNA, snRNA, third end of the intron. They will bind and they will act like a hook. With the help of the NURBs and the snRNA, they can break the bond between phosphodiester, 3,5-phosphodiester. Then they will act like a hook and with the help of that catalytic activity. So it will show the catalytic activity. They will break the bond between the 5'3" of the exon and the next exon. So the intron segment as such can be pulled out and the exons can be ligated together. So that snRNA plus small ribonucleoprotein snubs together, we'll call it as spliceosome. So with the help of spliceosome, the splicing, that means the removal of introns, is possible. And as soon as the introns are removed, the exons are united together. So we'll get a continuous stretch of RNA. One end it is with 5-methylguanosine and a continuous sequence of exon which ends in or terminates in poly A tail. So that RNA is called as mature RNA. And soon after the transcription, this mature RNA, that is in the form of mRNA, it can pass through the nucleopores and can assemble in the cytoplasm for the translation. So I'll read out the main points here. In eukaryotes, 3 types of RNA polymerases found in the nucleus. In addition to the RNA polymerase found in the organelles are involved in the transcription. That is RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. And RNA polymerase 1 transcribes the ribosomal RNA. RNA polymerase 2 transcribes the hnRNA, or we can say the precursor of mRNA. RNA polymerase 3 transcribes tRNA, along with that 5S rRNA and small nuclear rRNA. So RNA polymerase 3, 2 more functions are there. Its main function is tRNA transcription, and along with that 5S rRNA and small nuclear rRNA. Small nuclear RNA. Then the primary transcription has both exon and intron. Exon is expressible region or coding region. Intron is intervening region or non-coding region. And how this hnRNA is converted into mRNA? By the process, 3 steps: capping, tailing and splicing. So capping means an unusual nucleotide methylguanosine triphosphate is attached at the 5' end of the hnRNA and which act like a cap. so which will protect the RNA from enzymatic activity. Then tailing means adenylate residues of approximately 200 to 300 adenosine are added at the third end, and it is fully processed hnRNA, now called as mRNA. Simultaneously, the splicing process also will take place, that is, introns are removed by the process that is called as splicing with the help of spliceosome. And the whole hnRNA, by the time it will be converted into messenger RNA. So the difference between exon and intron. Exon is the region of gene which become part of the mRNA and code for different regions of proteins. Introns means the regions of a gene which are removed during the process of mRNA and they do not take part in protein synthesis. They are non-coding sequences which are found within the organism as a part of evolution. Then splicing means the process in eukaryotic genes in which introns are removed and the exons are joined together with the help of spliceosome to form the mature mRNA. Then what is the significance of the splicing? The split gene arrangements represent probably an ancient feature of the genome, because in ancient time the prominence was RNA genome, where the genetic material is RNA. So that time the climatic conditions were unsteady and it is not that much favorable. Always there was fluctuating climatic conditions, very hot, then suddenly very cold, and like that way. So that time the animals preferred a more modifying type of genetic material. So here RNA, if they are acting as the genetic material, means with the help of splicing they can modify every now and then and they can go for more adaptable genetic material to suit to the unsteady climatic conditions. So that is why the splicing mechanism is still retained within our body. So the presence of intron is reminiscent of antiquity, that means ancient characteristics, and the process of splicing represents the dominance of RNA world. That means we all were inherited from the RNA carrying ancestral form, where with the help of splicing they were able to adapt more and more in a better way to the unsteady climatic condition. That is why the splicing is very significant at present. So with this, we come to the conclusion of this module. Thank you very much.